understanding bacteria growth and recombination is very important to understand because from a doctor's perspective in a hospital, if you've got a patient with septic shock, you're going to have to understand the fundamental principles behind how these bacteria are evolving and responding to external stimuli, such as the antibiotics that you're giving, as well as what kind of growth phase they're currently in, to understand maybe what kind of dosage you're going to need to go with. And so what I'm going to do in this video is go over how bacteria are doing recombination, as well as why, and what their typical growth cycles look like. And so the goal of recombination is to get genetic diversity. And bacteria by themselves will perform something referred to as binary fission, in which they will do asexual reproduction. And while they're able to reproduce, this is not that great from an evolutionary perspective because you're not generating any genetic diversity. If one particular bacteria ends up being more fit to the environment, there's no way to get that. And so there's three methods of recombination. The first is referred to as transformation. And transformation is simply the incorporation of foreign genes into the host. And so it's pretty straightforward. With method 2, there is something referred to as conjugation. And conjugation is a bit more interesting in that it's kind of like bacteria sex, is the way I look at it. And what happens in this is there will be a male and a female, and the direction of gene transfer is only one directional. And so what will happen is we'll have some plus the male feeding its genes via some kind of sex pili to the female, the minus bacteria. And this is only happening in bacteria that are considered F positive. This is only for F plus bacteria. And what that means is that these bacteria have the sex factor plasmids. And the reason you need these sex factor plasmids is because you need to form these pili between these two individual bacteria cells in order to do this. And not all bacteria have this capability. So F minus don't. And finally, the other method of recombination that exists is referred to as transduction. And transduction requires a virus referred to as a vector. And these viruses, like bacteriophages, will transfer and shuffle genes among bacteria. And so all three of these methods get us the goal of genetic diversity in bacteria. And there is another kind of interesting note here, which is something referred to as transposons. And transposons are simply jumping genes. And these jumping genes, the reason they're called that is because they're segments of DNA that will insert and remove themselves from host genomes. And it is a source of genetic diversity that can sometimes prove beneficial to a host cell, for instance. And so if we were to examine a typical growth cycle of a bacteria, we're going to find 4 regions. We're going to have an initial region, a growth region, a stationary region, and then a death phase. And so if I number these just to keep things clear, we'd have 1, 2, 3, and 4. what's happening in each of these, so in case 1, the bacteria are in the lag phase, and in the lag phase, the baby bacteria, the new bacteria, are adapting to their environment. So if there was an environment in which lactose was abundant and no bacteria had the lactase enzyme present, this is that time period in which those bacteria will be recognizing that they need to start making lactase and beginning to digest it. And then what happens in this region 2 is something referred to as the exponential growth phase. And in the exponential growth phase, in the case of E. coli, they've got maximum substrates available to them so they can grow and divide as fast as they like. And we get rapid growth and proliferation of the cells. And what happens in region 3 is, like on the macro level, you've got so many finite resources, you eventually reach a point at which you're in a stationary phase. So growth stops due to resource limits. There's only so much glucose present in the solution, for instance. And step 4 is the kind of sad one, which is the death phase. And in the death phase, all the nutrients run out, and so no more substrates, and everyone dies. And so this is what we can expect to find in a typical growth cycle for bacteria, as well as how these bacteria are creating their genetic diversity. Let me know if you guys have any questions, and thanks for watching.